Presentation discusses how hair becomes damaged and the effects of hair dyeing, perming, and bleaching on amino acids and hair lipids. The presentation is being conducted by Vibui, Madeline Fleming, and Yali Chen.
Hair is important to human as it is associated with one's beauty in society. Thus, our group is interested in examining the hair damage when it undergoes chemical treatment. It was intriguing to look into how hair becomes damaged and the negative impact of dyeing, perming, and bleaching human hair, especially through the lens of amino acid and lipid. Our topic is also related to our class because we talked about amino acid and lipid before. Either you want to get your hair straightened or curl permanently. Both of the processes involve denaturing the protein in your hair by disrupting the disulfide bond found in the hair protein called keratin, which reshape the hair and forcing the disulfide bond to reform a different point. Hair shaft consists of 3 different types of bonds, which are hydrogen bond, salt, also called saline, and disulfide bond. Hydrogen bonding allows our hair to change shape temporarily and produce a flexible hold. Disulfide bonds are stronger and more permanent, thus requiring a strong chemicals to break them, such as aluminum dichlorate, a chemical that is used in perm. Keratin is high in sulfur and is the biggest component of the disulfide bond. The hair shaft with missing molecules of keratin often lack luster and are prone to breakage, and the broken disulfide bond within the hair weaken the shaft. Cysteine is the most crucial of amino acid for hair growth. It established disulfide bridge, a type of bond that plays a fundamental role in the folding and stabilization of protein structure, including the bonds within the hair shaft.
The hair shaft is composed of keratin, which is made up of amino acids. Amino acids are essential for healthy hair because they aid in hair growth, strength, and length retention, especially the amino acid cysteine, which forms disulfide bridges in keratin. Cysteine provides sulfur to hair cells, as well as the improvements of hair texture, elasticity, and strength, which can ultimately reduce the chance of hair loss. Hair lipids are what provide your hair with a smooth and shiny appearance. The hair lipid consists of 85% saturated and unsaturated fatty acids. The proper amount of hair lipid is what protects your scalp layer from overdrying. If the scalp becomes too dry, there will not be enough nutrition for the hair to grow. The lack of nutrition and water will also cause dandruff and even hair loss. The hair lipids can moisten the scalp and hair root, and it can also resist the invasion of harmful bacteria by forming a protective layer on the scalp.
This slide is about experimental design. In this experiment, hairs of other ethnic groups were not studied. There are 4 steps for the experiment. The first step is to purchase chemically untreated Asian hair. The second step is to cleanse hair by soaking in deionized water, then rinsing several times and air drying for 3 minutes. The third step is to undergo perming, dyeing, and bleaching. The last step is to observe and record the results. The figure consists of 6 parts. For part A, label the hair sections show how chemical treatments alter the amino acid composition compared to a control of chemically untreated hair. Changes may occur within the cuticle layer which was damaged in chemically treated hair samples. Part B, categorize hair by color. Part C, displays the level of amino acids. Chemically treated hair can be determined by the presence of cysteine, tryptophan, citric acid, and methionine. Part D shows modification in tryptophan and methionine. Part E shows the effects of citric acid. Part F displays the citric acid ratio of chemically treated hair. Results are statistically significant because the error bars of chemically treated hair samples do not overlap with the control. The data in Figure 1 suggests the use or overuse of chemically treatments negatively alter the amino acid and lipid composition of hair.
The research shows that disulfide bond of cysteine would form a disulfide bridge in keratin is harmed by oxidative stress caused by oxidative hair bleaching. Interestingly, chemical treatment of hair leads to oxidation in sulfur atoms in amino acid, causing the formation of cysteine, thus increasing the amount of cysteine present in the hair. This is opposite from what we would expect. However, this could be explained by the experimental error, time, and or the use of hydrogen peroxide to the hair as it has been altered by chemical treatment. Additionally, try to find an amino acid that is sensitive to reactive oxygen species or heat reaction was depleted by chemical treatment of hair. Methylene, an amino acid that is susceptible to the addition of oxygen to the sulfur atom, was found to be decreasing at a constant rate during hair bleaching. Furthermore, chemical treatment of hair causes a great number of hair lipids to be depleted, especially free fatty acid like erotic acid, behenic acid, lignoceric acid, nervonic acid, cerotic acid, and 18-MEA. Overall, the research demonstrates that hair amino acids and lipids are decreasing tremendously with chemical hair treatment, leading to the deterioration of fiber hair quality. This suggests that high levels of amino acid and lipid makes hair healthy, while decreasing the amount of amino acids and lipids in hair causes hair damage.
Our first exam question asks, what are the steps involved in the manipulation of chemical hair treatments and what do they do? The answer is the manipulations involve oxidizing and reducing steps that necessitate the use of reactive chemicals. Our second exam question asks, how does exposing hair to chemical treatment, such as dyeing, perming, and bleaching, cause hair damage? And this causes hair damage because healthy hair should contain high levels of tryptophan, methionine, and cystic acid, as well as cysteines, as it helps with the hair structure and promotes hair growth. Lipids are a critical component in the hair cuticle, and by introducing your hair to chemical treatments, it causes substantial amounts of these amino acids and lipids to reduce, which can result in damage and make your hair weaker.